Class 10 Biology, Chapter Heredity and Evolution, video number 2. In this video, I'm going to discuss about inherited traits. You'll be learning about Mendel's experiment, under which I'll tell you why Mendel selected garden pea plant for his experiment, a briefing about monohybrid cross, and the conclusion made by Mendel on the basis of the monohybrid cross will be discussed. As you all know, heredity is the transmission of traits from the parents to the next generation. And before we move further, we need to know the difference between characteristics and traits. As you all know, characteristics or features, they are the details of structure and working of an individual or an organism, and the distinguished expression of characteristics, that is what is called as trait. Like each character that you take into consideration, you will find many variations in that. So those expression of characters in the form of different variants is what is called as trait. Let us take an example. Height, if you take, it is a character. And height, if you ask, there'll be lots of variation in height. You find people being tall, they may be short, they may be medium height also. So the way these characters are expressed, that is what is called as trait. So being tall, short or medium height will be the trait of the character height. Or we can say, take another example as blood group. Blood group will be a character, and having A, B, AB or O group will be the trait. Okay, now when we talk about traits, traits are of two types. You have got acquired traits and inherited traits. So traits can either be acquired or inherited. The acquired traits, they are the traits which one gets during the lifetime. They are the traits that one gets during the lifetime as a result of practice or because of environmental factors. For example, being a good dancer or a singer or painting. These are all, or development of muscles. These are all acquired traits, and these traits are not controlled by genes. So they are not passed from one generation to other. Whereas inherited traits, they are the traits which are genetically determined, that is, they are under control or they are expressed by genes, and these are passed from parents to next generation. Examples of inherited traits, this includes eye color, hair color, skin color, and so on. So these all are examples of inherited traits. Now let us discuss about the rules of inheritance that Mendel had given. When we discuss about the rules of inheritance in human beings, one thing that we need to know is that for each and every character that an individual gets from the parents, because human beings are sexually reproducing organisms, so there are two parents involved, and both the parents contribute the genetic materials through those gametes that are involved in the process, right? So for each and every character that an individual has, both the parents will contribute their genetic material. So if suppose we take eye color, it's not just like, if you take this example that is being seen in the screen, you find that the mother has black eyes, father has blue eyes, and the child has black eyes. But having black eye does not mean that the child has not inherited any gene from the father. The child has inherited gene for this character or trait from the father as well as the mother. Now, what is going to be expressed in the next generation of the children, that is what Mendel had studied. Okay, so this is what Mendel had studied. Mendel, he worked out the rules of inheritance, and for his experiment he made use of the garden pea plant. He was the first person to study about how the traits or characters or variations are passed from one generation to other. So before we move further, you just have to remember that for any character or trait that we have, in case of, I'm talking about the sexually reproducing organism, both the parents that is the father and the mother, or the male and the female parent, both of them contribute their genetic material. Okay, so for each trait there will be two versions of genes in the child. One will be expressed and other may not be expressed. So how it is done, that is what was discussed or studied by Mendel. And before we discuss about his uh, experiment, let us first have a brief history about Gregor Johann Mendel, who is also known as the father of genetics. Genetics, as already told, it is a branch of biology which deals with the study of heredity and variation. So Mendel, he was born in a small family, in a farmer's family, and his initial education he had in a monastery. A monastery is a place where monks live. Okay, so he had his education in a monastery, and for his higher studies he went to University of Vienna, and while he was studying there he actually failed in an examination to get the certificate for teaching science and maths. He was a student of science and math also, right? So he failed in the examination, and after that he came back to the monastery and he started working as a monk over there. So while he was working in that monastery, he found that in the garden of that monastery this garden pea plant was being grown, and these plants that he observed, he found that those plants showed variations for several characters. Like for example, the characters that they showed variations. These are what is seen in the slide. These were the characters for which they showed variation. Like in terms of flower color, the flowers were either purple or white. The plants were either tall or short. So for several characters, they showed contrasting traits, and so he wanted to know what is the reason for this and how are these traits being passed from one generation to other. So he performed many crosses in this. We like in this chapter, you'll be learning about the monohybrid and dihybrid cross that Mendel had done, right? And then he came up with his conclusions, which are now taken as the laws or rules of inheritance. So let's know about that. And before we discuss about his experiment, we need to know what was the cause for selecting garden pea plant for the experiment. So the first and the most important reason why Mendel selected garden pea plant for his experiment was he could find, or he found, that the plant showed contrasting traits for several characters. Okay, in fact there were seven pairs of contrasting traits, that is the contrasting traits for seven characters were shown. So this was one of the main reason. The other reason for his selection of this particular plant was that the plant produced huge number of seeds each generation and had a shorter lifespan. These plants, pea plant is an annual, it completes its life cycle in one season. Okay, so because of this he could get quick results. Then the other reason was that the plants, as the plants had or produce bisexual flower, normally the plant underwent self pollination and self fertilization, but in a controlled manner with a quite easily cross pollination could also be performed. And after cross pollination, the hybrids that were produced were perfectly fertile. So because of these reasons, Mendel selected garden pea plant for his experiment. Okay, so here as you see, these are the seven characters for which contrasting traits or seven pairs of contrasting traits were seen, like round and wrinkle seed. This is a shape. In terms of seed color, it was either yellow or green. In terms of pod, which is a fruit of the pea plant, it was either flattened or it was constricted. The pod color was either green or yellow. No mixed other trait was seen. The flower colors were either purple or they were white in color. Flower position, it was either axial or it was terminal, and the plant were either tall or short. So this is the main reason why Mendel selected his this particular plant for his experiment. So now let us study about the experiment that he had performed. In this video, we'll be discussing about the monohybrid cross. A monohybrid cross, first let us know what it is. It is a cross or experiment that was done to study the inheritance of one character. How one character, like for example, as I said you, that this particular plant showed 7 characters where he found variations. So if suppose he was performing a cross, or if a cross is performed to study only one trait, say height, so only one character, not the other ones are ignored, and only about height, how height is being inherited from one generation to another. So if this is being done, then that type of cross will be said to be as a monohybrid cross. So in this cross, like let's take some examples. First we'll go with the height. So what Mendel did was he took pure tall plants, as you can see here. First Mendel, what did he do was, the parent that he made was pure tall plant and pure dwarf or short plant, and he crossed them. What is crossing? It is like in this, what he did was he made one plant as a female. Suppose this is made as a female, and how will it be made? I said you the flowers are bisexual, right? So from this particular plant he removed the stamens and like manually, and then he made it as female, and then he picked the stamen of the plant flower from another plant which was tall, and shedded the pollen grains on the stigma of this particular plant. So this is what he did. So he crossed, and he had taken pure plants. The plants were pure tall and pure dwarf. Now what is a pure tall plant or pure tall breeding plant? It is a one which will produce tall plants generation after generation whenever it is left for self-pollination and self-breeding. So if suppose, like here what is shown is cross-pollination, but if suppose the plant is left undisturbed, then it will undergo self-pollination. So on self-pollination, if the plant produce tall plants generation after generation, then it will be said to be as a pure tall, and the pure dwarf will be the one which produces dwarf plants or short plants generation after generation. So such pure breeds he took and he crossed them. Okay, he shedded the pollen grains from the tall to the dwarf one. And what he found in the next generation, which he termed as the first filial generation or F1 generation. So in the next generation, he got all the plants as tall. The dwarf trait was not at all seen. So he wondered, where is that dwarf trait? Because first generation he got all the plants as tall. So he wondered that where is that dwarf trait? And other thing that he had in his mind was that whether these tall plants are same as that of the parental plant. So for this he further extended his experiment, and what he did was he self-pollinated the tall parent plant, and that is he left the tall parental type for self-breeding. So since he had taken a pure line, he got all the plants, the progeny or the young ones that were produced as a result of self-pollination, they were all tall. Okay, so this is what he did. Then to confirm whether the F1 generation, the first filial generation plants were also tall, he left them also for self-breeding. So he left the plants for self-breeding. First he had done cross, now self-breeding. So when he left the plant for self-breeding, in the next generation, this is the F1 generation. So the next generation, which he called them as F2 generation, he got both tall and short plant, and that too in the ratio of 3:1. Okay, so this is what he got. He got these plants in the ratio of 3:1. So from this he made some conclusions. Now what were the conclusions? Let us see. What conclusion did Mendel make? I'm repeating that the first Mendel, he had crossed a pure tall plant with a dwarf plant. First generation he got all the plants as tall, and when he left the first generation plants for self-pollination, he got the plants as tall to short in the ratio of 3:1. From the experiment that Mendel performed, he came up with some conclusions. The first thing that he stated was that the fact there are these different traits, that is the trait of tallness and the trait of dwarfness, are controlled by discrete hereditary particles, which he called them as factors. And why he said so? Because he got only tall and short plants, like F1 generation all tall, F2 generation both tall and short. There were no medium height plants. So there were some discrete particles that were controlling the traits that he stated. And those discrete particles he called them as factors. Now, for giving a symbol to the factors, he made use of alphabets. Like for height, the alphabet that was made use was T. Okay, this was the alphabet that was made use of. Now, other thing that he stated was that in the first generation, when he got all tall plants, but when the first generation plants, when they were self-bred or self-pollinated, he got both tall and short plants in the ratio of 3:1. So from this he concluded that though the trait of shortness was not shown in the F1 generation, despite of this, he said that this F1 generation plant had both the factor for tallness as well as the factor for dwarfness. But the factor for dwarfness could not express itself in presence of factor for tallness. Okay, and such factors which express themselves in the F1 hybrid, that those factors he called them as dominant factor. So when a cross is done and a hybrid is obtained, the factor that expresses itself in the hybrid or in the F1 generation, he called them as dominant factor, and the one which remain unexpressed, in this case it is the dwarfness or the short trait. So the factor which controls this factor which remains unexpressed in the F1 generation, he called it them as the recessive trait or recessive factor. Okay, so the factor was called as recessive factor, and the trait that is controlled by this factor is called as a recessive trait. So this is what he said. Now for giving like symbolizing these factors, he made use of alphabets, as I said you. Now for a dominant factor was expressed by capital letter case, and the recessive one was expressed by small letter, right? Now for height, T was a symbol that was made use of. So he said that the F1 generation plant had both the factor for tallness, because that was being expressed, and the factor for dwarfness. And from this he also concluded that in an individual, that is F1 individual, which is showing the trait of tallness, in an individual, factors are always present in pair. Factors are always present in pair. Now this pair of factor may be either similar or dissimilar. Here F1 generation plant, they are having dissimilar factors, a factor for tallness and a factor for shortness. But in the pure breeds, if we go with the pure breeds, like pure tall plant when he took, which was giving only tall plants generation after generation whenever it was selved. So for this particular parent, the genetic trait factor or genotype of this particular parent, according to what Mendel concluded, should be two factors for tallness, and that of a dwarf plant, because dwarfness was not expressed in the F1 generation, means dwarf is a recessive trait, so he made use of 2 small t. It's 2 small t because it's a pure breed, and every generation it's going to show the same trait. In fact, a recessive trait, the one which is not expressed in the F1 generation, is called as a recessive trait. They are the genotype of this particular character which is recessive. It will be having only the recessive factors. So the genotype of a dwarf plant will be 2 small t's together. Okay, so the factors, they are present in pairs, and this factor can either be similar for a pure, as in case of pure tall plant, or pure dwarf plant, or dissimilar as in case of a hybrid, which will be a factor for tallness and a factor for dwarfness. He also stated that these factors which are present in pair in the individual, they get separated at the time of gamete formation, because it is sexual reproduction, and you have also learned that process involves gametes. So the factor which is present as a pair in an individual will get separated at the time of gamete formation. Okay, they get separated at the time of gamete formation. This means that each gamete gets only one factor for each character. Individual will have two factors, that is factors in a pair, but the gametes get only one factor for each character. Other thing that he said, that these factors which get separated at the time of gamete formation, they pair up again after fertilization. Means a factor like the pure parent which is tall will produce gametes which will have only the factor for tallness, and the parent which is dwarf and pure, it will be producing gametes which will be having a factor for dwarfness, that is small t. And when these gametes, because sexual reproduction involves fusion of gametes, so when these gametes fuse, the resultant F1 generation will have a factor for tallness and a factor for dwarfness. So the genotype of the F1 generation plant, which is which is a hybrid tall, will be a capital T, that is a factor for tallness, and will have a factor for dwarfness, right? Then as I said, that these factors, they get separated at the time of gamete formation, and again he stated that these, when they pair up again after fertilization, and fertilization is random, right? So let's see like how this happens. Here, as you can see in this slide, we'll start with this same explanation once again. Mendel had taken pure tall plant and pure short plant as a parent, and as already stated, the tall plant will have two factors for tallness, and the short plants will have two factors for shortness. Now since it is, he had crossed them, and it's a sexual reproduction, it involves gamete formation. So the gamete that the tall parent plant produces will always carry only a factor for tallness, and the gamete that the short parent produces will have a factor for shortness. Now the factors, he stated that this will again pair up, and this pairing occurs during fertilization, and fertilization is random. Any gamete with any kind of combination can pair up with any other gamete, but in this case you find there's a the male gamete, if this is a male parent, then it's carrying a factor for tallness, and the factor for dwarfness is being carried by the next the gamete produced by the other parent. So when they pair up, the resultant F1 generation plant will have a factor for tallness and a factor for dwarfness. Since the factor for tallness is dominant, it is, the plant is going to be tall, and factor for dwarfness, that is small t, is recessive. This is going to be hidden, and so the plant in F1 generation will be purely it will be a tall plant. Now what the next thing that Mendel did was, when he got the F1 generation plant as all tall, he wanted to know whether these tall plants were the same as a parental type. So he self-bred the F1 generation plant. That is, he allowed the pollen grains of the present on the flower of the same plant to fall on the stigma of the same flower. So when this happened, like again it is sexual reproduction, so the gametes will be involved, and there are two parents, the stamen and the carpel. So that's a male and the female part of the plant. So here when we do a self, there'll be the male part of the flower will have the genotype as capital T and small t, and since it is self pollination, the female part of the plant also will have a genotype of capital T and small t, that is a factor for tallness and a factor for dwarfness. As already stated, that these factors again will get separated at the time of gamete formation. Gametes get only one factor. So this particular plant that you see, it produces two types of gametes. 50% of the gametes will carry a factor for tallness, and 50% of it will carry the factor for dwarfness. Similarly, since it is a self-breeding and self-pollination and self-fertilization occurring, the other parent, that is, will have the same genotype, and it also will produce gametes, where the gametes will carry 50% of them will carry the factor for tallness and 50% a factor for dwarfness. Now he stated that these gametes will fuse again, and pairing of the factors will take place again. But then this is a random fertilization. So this pairing is random. So if suppose a gamete carrying the factor for tallness fuses with the other gamete which also has got the factor for tallness, then the resultant progeny, that is the F2 generation, this will have two factor for tallness, and so the plant will be tall. Now when the gamete that's carrying the factor for tallness fuses with a gamete that's carrying the factor for dwarfness. The resultant zygote will have the factor for tallness and one factor for dwarfness. And as tallness is a dominant trait, the individual that is going to develop when the seeds were sown, the individual that developed will be a tall plant, right? And this is because like we have to go with all the possibilities, like a possibility like a gamete fusing with the other gamete carrying the factor for tallness, a gamete with the factor for tallness fusing with that with a gamete which has a factor for dwarfness. So this shows that fertilization is random, and so we have to go for all the possibilities. Now the next possibility is if suppose a gamete, because this particular plant will produce two types of gametes. So we have gone with the possible modes of fusion that for this particular gamete. Now let's discuss about the gamete which is carrying the factor for dwarfness. So a gamete which carries a factor for dwarfness, that is small t, when it fuses with that of a gamete which carries a factor for tallness, the resultant zygote will have, or the individual of the progeny that develops will have a factor for tallness and a factor for dwarfness. And always remember that when you write these factors in a pair, the dominant trait has to be written first and the factor which is recessive has to be written next. It should never be written the way like this, what is being written on the screen. That is a recessive factor followed by a dominant factor. So the resultant zygote which is produced, or the individual that is produced, will have a genotype of having a factor for tallness and a factor for dwarfness, and so it is going to be a tall plant. Now if suppose, because fertilization as said is random, this gamete which is carrying the factor for dwarfness fuses with that of the factor, another gamete produced by the other parent carrying the factor for dwarfness, then the resultant individual which will be produced will have a genetic makeup of two factors for dwarfness. And since it's having two factor for dwarfness, the resultant plant will be short or it's going to be dwarf. So this is how he this fertilization results in, this random fertilization results in all these different possibilities. And when you see, you find that the external appearance wise, like that's what's called as a phenotype of the progeny of F2 generation, you'll find that there are three possibilities where you find it's tall and there's one of short. So the F2 progeny produces tall and short plants in the ratio of 3:1. It's not that there are 4 plants produced. If suppose a cross was like when the plants were allowed for self-breeding, and then when the fertilization happened and when seeds were produced, Mendel might have counted the seeds, and so suppose the number of seeds produced was 100, then of the 100 seeds when it was sown, 75 seeds grew into tall plants and 25 seeds it grew into short plants or the dwarf plants. So the ratio in which tall to short or dominant to recessive traits that appear is 3:1, and this 3:1 ratio is called as the monohybrid ratio. It's called as monohybrid ratio. This a monohybrid ratio is a ratio of F2 progeny that is formed when a monohybrid cross is done and the F1 generation plants are made to self pollinate or self breed. So the ratio that is obtained is in the F2 generation is what is called as a monohybrid ratio. And the phenotypic ratio, there are two terms, phenotype and genotype. So phenotype stands for the external appearance and genotype stands for the genetic makeup for that particular trait. So the phenotypic monohybrid ratio is 3:1. That is 3 tall to short ratio, it is 3:1. And when we go with the genotypic ratio, you find that there is two capital C's, that is individuals with two factors for tallness, so that is 1. Then there's you find 50% of them have a factor for tallness and a factor for dwarfness or shortness, so that is 2. And then 25% of them will have two factors for shortness. So this 1:2:1 ratio is a genotypic ratio. Okay, this is a genotypic ratio. It's on the basis of the genetic makeup of the individual. So genotypic monohybrid ratio is 1:2:1. So this is about the hybrid cross, and here this is a tabulated form where you can see clearly that how this cross was done. Tall plants were crossed with the dwarf plant. The F1 progeny were all tall, having a genetic makeup of a factor for tallness and a factor for dwarfness. Then they were allowed to self-pollinate, and then every time you, as already discussed, that the sexual reproduction will involve gametes. So since it is a self-pollination, the male and the female of the flower will be having the same genetic makeup, and so they will produce like this. Since the genetic makeup is a factor for tallness and a factor for shortness, 50% of the gametes will carry a factor for tallness, 50% a factor for dwarfness, and this will be same for both the cases. And when fusion occurs, when you find the fusion occurs, you find that 75% of the plants grew into tall and 25% of it grows into a short plant. Okay, so that's the monohybrid ratio, and this is not only in case of height. If suppose another cross is done, say taking the character of flower color, as already said in the beginning that the plants, the garden pea plant showed two types of they produced two types of flowers. Some of them were producing flowers which were purple in color, and some of them were producing flowers which were white in color. So when Mendel performed this experiment, taking pure lines of purple plants producing purple flowers and those producing white flowers in the and when he crossed them in the F1 generation, he got all the plants producing flowers which were purple in color. And then when he allowed those F1 generation plant to self-pollinate, then he got the F2 generation where he got plants producing purple flower and plants producing white flower, again in the ratio of 3:1. So that since the F1 generation plant, there it was only flowers with purple color, this purple color of the flowers was said to be as a dominant trait, and the white flower, because white color was not expressed in the F1 generation, that's why the white color was referred to as the recessive trait. It is the recessive trait, and the factor that controls this white color will be called as a recessive factor, and the purple, which is shown in the F1 generation, so the factor that controls expression of it, that is said to be as a dominant factor. Okay, let's take another example. Here this is an example of a cross between the plants producing round seeds with that of the plants producing wrinkled seeds. So again, as said, because in like when it was a cross in the F1 generation, he got all the plants which were round. Now when, and here again you can see here like those if we go with the genetic makeup, since it is a pure breeding round seeded plant, the genetic makeup of it will be having two factors for round shape. Okay, so 2 capital R is the symbol that is made use of, and in case of wrinkle seeded plant it will be 2 small r's. Now, as it is sexual reproduction, it involves gametes. So the gametes that this particular parent produces, all the gametes will have the factor for round shape, and the other parent will produce the gametes which will be having the factor for a wrinkled shape. Now, when these factors fuse, so fertilization will be following after gamete formation, fertilization will take place. So when these gametes fuse, the resultant F1 generation plant or progeny will be having a genetic makeup of a capital R and a small r, that is a factor for round shape and a factor for wrinkle. And since round shape is a dominant trait, it is round shape that is expressed in the F1 generation. When this F1 generation is allowed to self-breed, as shown over here, you see here what is the progeny that one gets in the F2 generation. So since it is self-breeding, the genotype of male and female will be the same, and there will be similar kind of gametes produced by each of the parent. That is, the parent will produce gametes which will be having either the factor for round shape or wrinkled shape. Similarly, the other parent also, this is a male, so there will be a round shape gametes carrying the factor for round shape and the gametes carrying the factor for wrinkled shape. Now, as fertilization is random, we have to go with all the possibilities. So when a gamete carrying the factor for round shape fuses with the gamete carrying the factor for round shape, the genotype will be 2 capital R and the shape of the seeds will be round. Similarly, when this gamete may also fuse with another gamete carrying the factor for wrinkle. So the genotype of it will be capital R and small r, and so since round is a dominant factor, round R is a dominant factor, the resultant seed in the next generation will be all round. Similarly, there is another possibility that if suppose a gamete is having the factor for wrinkled shape and this fuses with the gamete which is having the factor for round shape, then the genetic makeup will be of the zygote and the individual will be having a factor for round and a factor for wrinkle, and as round is dominant factor, the phenotype of the seed will be round because round is the dominant factor. And if suppose fertilization is such that the gamete carrying wrinkled shape, factor for wrinkled shape, fuses with another gamete which is having a factor for wrinkled shape, then the genetic makeup of the individual will be, for shape of the seed will be two factors for wrinkle, and so its shape will be wrinkle. So when we see the ratio for round to wrinkled, that is dominant to recessive, again over here we see that the ratio is 3:1. So always in any kind of monohybrid cross you get a ratio of 3:1. So on the basis of entire of the experiment and observation that Mendel made, he finally concluded. So that is, his conclusion is summarized over here. What Mendel stated, that the traits are inherited in the form of discrete units. Okay, so there are discrete particles which control the expression of traits, and they are passed from one generation to other. Right, they are passed from one generation to other. Why discrete? Because there is no blending. When we took the experiment as a tall plant being crossed with a dwarf one, we got tall, and second generation we got the plants both tall and short, but there was no medium height. So there's no blending of these factors or discrete. They are discrete units and the factors don't blend. Okay, then he said that in an individual, he said that these factors in an individual, they are always present in pairs. And this pair of factor can be either similar, as you can see in this, that there may be for height there may be two factors for tallness, two factors for dwarfness, or the factors in a pair may be dissimilar, also having a factor for tallness and a factor for dwarfness. Right? Then other thing that he stated was the factors which are present in pair in an individual, they get separated at the time of gamete formation, and that is each gamete will get only one factor for each character, only one factor for each character. Okay, and other thing that he said that these factors, they again will pair up. So they are going to pair up again, and this pairing will occur during fertilization. So the factors pair up again during fertilization, and fertilization is random. That's why we have to go with all the possibilities. Any gamete produced by one parent, we have to go with what will be the possible result if it fuses with another gamete, any of the gamete produced by the other parent. Okay, so this is the conclusion that he made. So during reproduction, what he stated, that the inherited factors that determine the traits, they get separated when gametes are formed, and they pair up again, that is during fertilization, and everything of this, that is separation and again pairing up, is a random one. So there's reshuffling of genes, that is what is said during gamete formation and during fertilization. Now one of other important point that he stated, that in a hybrid over here we mean the F1 generation. So you find that a factor that is expressed, because hybrid will be expressing only one trait. So the factor which is able to express itself in a hybrid, it's called as a dominant factor, and the trait which is being controlled by that particular factor that is expressed in a hybrid, that is called as a dominant trait. Whereas the factor which remains unexpressed in a hybrid is said to be as a recessive factor, and the trait that remains unexpressed in the hybrid or in the F1 generation is called as a recessive traits. So these were the conclusions made by Mendel. Okay, so these were the conclusions that Mendel had made. So here the screen, you see there are all the dominant traits and the recessive traits are being shown in terms of seed shape. Round is a dominant trait, wrinkled is a recessive trait. In terms of seed color, yellow is a dominant factor or a dominant trait and green is a recessive one. Flower color, purple is dominant and white is recessive. Flower position, axial is dominant, terminal is recessive. And similarly, plant height, tallness is a dominant trait or the factor which is responsible for this expression is a dominant factor, whereas short is a recessive. So these are all examples. There were 7 characters for which Mendel found contrasting traits. So these are all those contrasting traits that he found to be dominant and recessive. There are other examples also of dominant and recessive traits, like as in the beginning we had discussed that earlobes, when you see there are 2 types of earlobes that people may have. The earlobe may be either free, okay, or it may be attached. So you must have seen most of the people having a free earlobe, and free earlobe is a dominant trait, and having an attached earlobe, that is a recessive trait. Similarly, in terms of eye color, you find that black eye or brown eye is a dominant trait, and the one that is recessive is a blue eyes. So that's about the monohybrid cross. In the next video, I'll be discussing about the dihybrid cross and what conclusion Mendel made on the basis of the dihybrid cross.